Well, welcome back, everyone, and thanks for coming. This week we've been talking about the case control study design. We've been talking about different aspects of that type of study. We've been talking about different ways to select controls, different ways to interpret and measure of associations that we might capture from such studies. Now I want to give you the opportunity to read and critique a case control study. So I've asked my friend and colleague, David Hunter, professor of epidemiology, here to come and to present a paper to you, give you a little bit of background, and present some questions that you're going to be asked to discuss on our discussion forum. Later on, David's going to come back and give you his solutions or his answers to those questions, as hopefully well incorporates some of the comments that you've made. But David, before you do that, I have to tell you that David wears another hat at this school. He's our Dean for Academic Affairs. He's the reason Marcello and I are here doing this course. It was his vision, his idea, to try to promote a course from our School of Public Health as one of the first edX courses offered through Harvard. So David, I'd wonder if you could give us some reason for why you did this to us, and what's your vision for courses like this and how it meets the mission of our school. So David, I'm going to turn the floor over to you.
So Fran, thanks so much. And to answer your question, as public health researchers and teachers, we want to get the word out about our set of methods to as many people as possible. That's definitely part of the mission of the school. We've always had a faculty who have done a lot of interactive teaching around the world, but there's only so many days in the year. And so people like you, Fran, you've done a lot of work in Argentina, for instance, and that's an absolutely wonderful opportunity for us to learn from our colleagues in Argentina, as well as participating in teaching with some of their students. But it's inevitably limited to the amount of time that people have to travel long distances. And so we're very excited about the fact that with edX, we can scale this up to thousands or tens of thousands of students. And I'm very grateful to you, Marcello, your TAs, the whole edX team, for taking on this task of leading the way, conducting the experiment, showing us how this is done. And we're very hopeful that faculty members from other departments in other disciplines will pick up this lead, and we'll be offering more courses in more public health disciplines in the next years as edX expands. So with that, I want to return to the subject here in week 8, and we're going to break this into 2 chunks. The first chunk, I will talk about some of the fundamentals of genetic epidemiology. I want to really stress that though there's a lot of jargon in the papers here, there's a lot of specialist knowledge. This is really no different from any regular case control study design and the application of regular epidemiologic methods. And then we'll take a break, and then I'll come back to talk about a specific paper and pose some specific questions for the students. So genetic epidemiology has been a discipline within epidemiology for many, many decades, but it's really been revolutionized in the last 10 to 20 years, really in the last 10 years, because molecular methods have become available that have really very much changed the way genetic epidemiologists do their work. In the pre-molecular era, most genetic epidemiologists were studying families or ways in which diseases traveled in families in order to reach conclusions about what the genetic and inherited component of disease is. And that was a very productive era, particularly for people who have a very, very severe or marked family history of a specific disease. It was often possible to decide whether that was on the basis of an inherited genetic component or something else, an environmental factor. But it was always quite limited, because even if we could establish that this was likely to be an inherited genetic disease, we didn't have the tools to discover which gene on which chromosome the molecular variant that was giving rise to the disease lay. And the molecular biology revolution in the last 5 or 10 years has given us the opportunity to actually nail down the specific variants, or at least the specific parts of the genome in which those variations are causing disease. So genetic epidemiologists still study families, particularly in the context of a very high burden of a specific disease in families. We study more than diseases. We study characteristics and traits that might be disease-related, such as height and weight, but are not specifically diseases. And we do that often in families. But in the last 5 or 10 years, a range of techniques have become available that allow us to do this in regular population-based study designs, notably case control studies or case control studies nested within cohort studies. So I'm going to show a series of slides from an article here that's available at the New England Journal of Medicine website. If you care to read the article, it muses not just about the design, but also about some of the applications of this knowledge. But the fundamental design here is something called a two-stage or a multi-stage genome-wide association study. And the basis of this is that new tools became available just 5 or 6 years ago in which we could test for the first time at initially 100,000 and then 300,000 and then 500,000 and now up to about 2.5, even 5 million specific genetic variants in a single DNA sample from a single case or a single control. We could actually establish what the precise variant was at each of those points in hundreds of thousands of places in the genome. And this revolutionized the way that we do genetic epidemiology. The problem was that this technology is relatively expensive. It costs many hundreds of dollars to do these very large-scale tests on an individual subject. And so the two-stage design acknowledges the fact that the first of these stages is relatively expensive per subject, and so we're inevitably limited on the number of cases and the number of controls that we can do this genome-wide experiment. We then take a handful or hundreds or thousands of the variants, I'll show how those are selected, and test these in a second stage, a much larger study at lower cost per subject. But this way we can do a genome-wide study, do replication studies in as many cases and controls as we can, and then blend the results to achieve a final summary statistic or p-value. So the genome-wide study, again, we can talk about single nucleotide polymorphisms, but just think of these as genetic variants. It's the most common type of variation in the genome. And we take the results of the first stage and take a handful of the results into the next stage and test for association. So all you need to know about genetics, and again there's a lot of jargon when you come to the paper, is that at each point in the genome we've inherited a copy of a particular genetic variant from our father, a copy from our mother. So we had to consider two options or possibilities at each point in the genome. And since we have two possibilities at each point, we can be a so-called common homozygote. So homozygote means that the variant is the same. And if we're common homozygote, that means that we have the variant inherited from our mother, inherited from our father, that most people in a particular population have at that point in the genome. Alternatively, we could be a variant homozygote, and so that means that we've inherited the less common variant, but we've inherited on both of the copies, one from mother, one from father. Or we can be heterozygote, which means that we have one of each. We have the common variant, we have the less common or rare variant at each point in the genome. And so what we do with a genome-wide association study is take these gene chip devices, which can measure many hundreds of thousands of these variants in a single sample. The people who made these devices have designed these across all of the chromosomes in the genome to be relatively equally spaced, so we can do a genome-wide study. And then at each point in the genome, we assess for each of our cases, whether they are common homozygote, heterozygote, or variant homozygote. We do this for each control case. In a large series, we do this for controls, people without disease or people who haven't developed disease at this point in their lives. And at each variant, we then can assess whether the variant or the homozygous variant genotype is more or less common in the cases than the controls. We then can assess this across all of the chromosomes depending on how many variants on the chip, but many hundreds of thousands. And what that leads us to is a so-called Manhattan plot. We've never had this sort of scale of data in epidemiology before. Most of the examples that you've studied, we're looking at a single exposure or maybe a few dozen exposures. But we tend to be looking at each exposure at a time, doing a chi-square and examining that association. Here, suddenly, we're looking at hundreds of thousands of variants, hundreds of thousands of genetic exposures, and hundreds of thousands of chi-squares. So the first problem was, well, how do we represent these in a way that the human mind can understand when there's so much data? And some very smart people in the Boston area actually came up with the graphic form of a Manhattan plot, which is illustrated here. So each of the chromosomes are listed here in a different color, and each dot here represents a p-value. So you can hone in on a specific genetic variant in each of the chromosomes. And it's a Manhattan plot because the very, very small p-values, the p-values with a lot of zeros in front of them, are actually high on the y-axis here. So these are the more extreme p-values, the p-values with the most number of zeros, the smallest p-values, and so the most highly significant in this initial pass through the genome. Now again, this was generated from a large genome-wide study that's expensive per sample, and we can't afford to do that on all of the cases and controls we might be able to assemble in the epidemiologic studies. So what we do is we skim off the most extreme p-values here and then use lower cost per sample techniques to see for as many of the initially extreme associations, how many of them replicate in a larger series. In the replication phase, we assemble as many independent case control studies as we can. We use lower throughput, but also lower cost per subject devices, and we just repeat the exercise. But this time, we're not looking genome-wide. We're looking at the places in the genome where we got these initial hints because we had variants that had extreme p-values. And so again, at each variant, though, we sort people into the common homozygote, heterozygote, variant homozygote, and we're looking for variants that are more common or less common in the cases versus the controls. And so what that leads us to is tables. So we use our lower throughput devices. We've got statistical tables and summaries of the data. Now we're not looking at hundreds of thousands, so we can actually usually assess these in more conventional tabular form. And we're looking for variants that replicate, and then we basically wind up with a summary across all of the studies. So we basically fuse the results of the first genome-wide stage with the replication stage. And if we're lucky, we will wind up, represented by the arrows here, with a small number of genetic variants at very specific points in the genome, at which the variant is more common or less common in our series of cases than it is in our controls. So these then are the markers in the genome where we think that there's a variant that is causally associated with the disease. Again, it could be a risk factor, it could be a protective factor, and then we basically go and look in those areas, what genes are in those areas. One sort of subtlety here is that the markers that we put on the initial SNP chips and the markers that we test for replication may not be actually the variants that are causally associated with the disease. They're just markers, and they may just be highly correlated with the true causal variant that we believe is in the neighborhood of the initially associated variants. So there's another step here, which is then looking at fine scale at the genetic variation at each of these points to try and discover what the causal variant at each point in the genome is. But that's the overall summary of genome-wide association studies. And what I want you to do when you read the paper is just try and screen out a lot of the genetic jargon, try and screen out a lot of the specialist genetic techniques, and just focus on the fact that what we're doing here is essentially a generic case control study. We're looking for things that are more or less common in the cases than the controls. And the real difference here is we're doing this in an industrial scale with hundreds of thousands of potential variants. And so that has implications for how we interpret the data. And we'll return to that when we look at the specific paper.